Hello, everyone. It is 10/9, 1:31 p.m., October 9, Wednesday. You had your exam today. Let's see. I'm being recorded. Okay, let's share. If you remember last time, we stopped by we stopped at the inhibitors. What I mean by inhibitors, it means the enzymes. I have to, in the old days, they used to give me, Delta College gave me a laptop that I could draw, and on the white area here, you guys can see, I could draw and do things. But anyhow, that's past, but I have to use my hand. So imagine this is an enzyme, right? And this is the substrate. This is the substrate. This is the enzyme. And now this place of that enzyme is the active site. You remember we talked about all of this. So this is the active site. This entire thing is the enzyme, right? And this is the substrate, comes in here. The shape of enzyme changes from last lecture. I forgot to put it in the YouTube. I will do it today after class. I should write it down on a piece of paper. Come on, let me put lectures on YouTube. Okay, put lectures on YouTube. Okay, so the shape of the enzyme changes. The molecule either come together or the molecules separate. All sort of possibilities. Do you remember the example of priest and the judge I talked about? Okay, but anyhow, now we are talking about an inhibitors. This is the enzyme. This is the active site, right? Another molecule will come and sit here on the enzyme, on the active site, or maybe others, which we talk about that. But for now, let's say it sits on the active site. Hey, the substrate cannot go in there anymore. This is an inhibitor. This is I hope I made sense. Now let's see what it says. Inhibit a substance that binds to the enzymes and decrease the enzyme activity. Okay, so that's, oh, go back. So it decreases the enzyme activity. And competitive inhibitors, it means competes with a substrate for active site. You remember I showed the rest of my arm too. I didn't stop the image right here. I went right here. So if the inhibitors sits here, that would be the active site. However, non-competitive that's called the competitive inhibitors. Non-competitive, it binds to the enzyme at a site other than the active site. For example, if this sits here and the active site becomes like this. So when the active site becomes like this, I cannot fit it in. This substrate does not go in. It's too tight, for example. Okay, guys, I hope I'm making some sense. So causes shape change right here. This inhibitor sits here, it changes the shape. It's too narrow. Too narrow for this substrate to goes in. You will see some pictures. And the enzyme unable to bind to the substrate right here. So this is a substrate. This is competitive inhibitor. Right? And this is non-competitive inhibitor. They call this site allosteric site. There is allosteric site here too. Most all enzymes, again, I didn't take a class by name of enzymology. There are classes offered by name of enzymology, but I didn't take it. But as much as I studied in my other classes, oh God, name a class that didn't talk about enzymes or substrate. But anyhow, that is called allosteric. Inhibits change shape of the enzyme so it cannot bind to substrates. So allosteric, let's focus on the allosteric sites now. You all know what allosteric site is right here. Right here. This is allosteric site. This will come up again later on during semester. I'm hoping you would understand it now. So when we talk about DNA and all of the enzymes involved with DNA, you will know what I'm talking about. Okay, allosteric enzymes exist in active and inactive forms, of course. This is active form of enzyme. This is inactive form of the enzyme. Right here. Okay, let's move. Most non-competitive inhibitors binds to allosteric sites, chemical on and off switch, which you will see that later on during semester. This concept does not stop here in the semester. It will come up again, and I will not go into details as much as now. Okay, but anyhow, allosteric inhibitors binds to allosteric site and reduces enzyme activity. We already know that. Okay, allosteric activators bind to allosteric sites and increase enzyme activity. So you have an inhibitor, right? In this case, this is inhibitor. There are other allosteric sites, that's the next thing, will bind here and make the enzyme work faster, better. The enzyme needed. I hope I'm making some sense. So this sentence is kind of, I would put 3 stars next to it, binds to allosteric sites and increase enzyme activity. What? Like what, Amir? Cofactors and coenzymes. Very good. Your textbook broke them down to 2. Good job. So cofactors, assisted enzyme, they are like co-pilots, co-worker. You've heard of those terms. So cofactors assist enzymes can be metal ions like zinc, molybdenum, manganese, there are iron, there are few of them. And are often found in the active site. They can be found in active site. So they are cofactors. Coenzymes, on the other hand, they are coenzymes. They are cofactors that are non-protein organic molecules. So the main difference between cofactors and coenzymes are the coenzymes are non-metal. They're non-organic protein molecules. They're organic protein molecules like vitamins. You remember? For this exam, I gave you 1 calorie is equal to, you increase the temperature of the 1 liter of water, 1 Celsius, so on and so forth. And I gave you 1 gram of carbohydrates, 1 gram of protein, 4 kilocalorie, 1 gram of fat, 9, 1 gram of alcohol, 7. Remember all of that? Vitamins, how much energy would I give you? 0. Trick multiple choice question. 0. Vitamins do not give any calories. Water does not give any calorie. It has to be those 4 that gives you calorie. I hope I'm making some sense. Okay, so vitamins, let's see. Metabolism, total of all chemical reactions. I talked about that. Anabolic reactions, anabolism. Oh, he did not go into coenzymes. That's okay. Vitamins, you will see, no, you will not see. I was going to talk about cellular respiration. There are some vitamins involved for cellular respiration. Anyhow, you will not see it. I guess this class does not go into those details. That's fine. Total of all chemical reactions carried by an organisms. We talked about this anabolism and catabolism at the very beginning of the very first PowerPoints. I talked about this. Anabolic reactions, anabolism, expand energy to build up molecules, building up muscles. So I take protein shakes. That's anabolism. Anabolic steroids, you've heard of them. they build up muscles. And then you have catabolism. Catabolism breaks down molecules. All of these molecules I'm drinking in this shake, it's not same molecules as my body. It goes inside of my body, it breaks open, and then they come together, anabolism, the way that my body needs them, not the way they are here, not the way the plants were using them or animals were using them. Okay. I hope I'm making a harvest energy by breaking down molecules. That's another thing. When I drink this, I get energy out of it because there are molecules here that when they break down, they release energy, which it will come up next chapter. Biochemical pathways, yes, reactions occur in a sequence. You will see that next chapter, and I will start next chapter today. Okay, product of one reaction is the substrate of the next reaction. You will see that again. Many steps takes place in specific organelles. Yes, you will see that. So this is a biochemical pathway. This is a cell membrane, right? You have enzyme 1, enzyme 2, enzymes 3, enzyme 4. These are phospholipid molecules. So what will happen? The initial substrate, right, comes in here. And then this product is made. This product goes in here. Then this product, look at the shape of the enzyme. Look at the active site. It's a square. Look at the active site. As the active site look a V, the triangle fits in there. Okay, look at the active site. It's a half a dome. Am I making sense? These are the substrates. And these are the enzymes, and look at them. They're all different. All of these enzymes are different. The shape, the name. And then, of course, this enzyme makes, this is a nice one. It goes in here. That's a substrate for this one. And the product of enzyme number 3 is the substrate for enzyme number 4. And number 4 puts out this end product, which the cells can use it. Okay, feedback inhibition. I talked about it a little bit in the past, just like a thermostat in your home, but it's coming up here now. End product of a pathway increase in concentration of its synthesized, more product increase probability that it binds to an allosteric sites on the enzymes in the pathway and causes it to change, and it cannot be bind normal molecule. Yeah, you will see that. It should come up. It will come up. Shuts down the pathway so raw materials and energy are not wasted. So what this thing is saying, let me give you an analogy. At your home, you have your thermostat. Nowadays, the temperature is okay, but a couple days ago, it was hot. So you set your thermostat for 80 degrees. So as soon as the temperature reached 80, the thermostat went off. When the temperature went back to 84, 83, 82, the thermostat was clicked on. That is and when the temperature reached 79, the thermostat went off. On, off, on, off, all day. Right? So that is called negative feedback inhibition. Negative. You have two. We have positive and negative feedback. This is an example of negative. The thermostat is off, on, off, on. That's negative feedback. What is Amir? What is an example of a positive feedback? Positive feedback, for example, you have an avalanche, a little snowball starts from top of a mountain, and that little snowball gets bigger and bigger and bigger and bigger and bigger and bigger. That is positive feedback. What is an example of that in our body? Females who are giving birth. At the beginning, they release a little bit of oxytocin. As they push and the muscles contract of the uterus, they release more oxytocin. When you cut yourself, a few the blocks, the coagulation factors will come to your site of the cutting, right? As time goes on, more and more and more and more come, so it will block. There are a few examples in the body for positive feedbacks, but the examples of the negative feedback are much more. A few examples here and there positive, but negative feedbacks, there are more. Feedback inhibition, again, same example. The circle comes and triangular and the square, and this is the end products. At the end, this one, it will go back and attaches to the allosteric site. You remember that? And the enzyme, the active site, narrows down. So this does not fit in here anymore. It means cell, enough of this molecule. We don't need it anymore. Stop. Just like a thermostat. Your cell made a whole bunch of these green oval shape product. You will see examples. Hang on. Okay? I hope I'm making some sense. Okay, that's the end of the chapter here. And I guess one of your classmates goes farther and study, and study the rest of this, I guess. And I said, do not worry about these. But anyhow, if you want to read those, please read them. And then I'm done with that chapter. Let's go. I hope you guys can see this. Okay, great. So chapter 7, these are overview of cellular respiration. We did it in the lab. And they say if you put lime or lemon, of course, if you put it in a food, you destroyed it. It has no activity. If you cut it and put it in the environment for more than 40 minutes, it's not effective. So I'm trying to finish my drink in less than 40 minutes. I don't know how long we've been at it, not that long, but what I put in here, you guys, you might find it beneficial. You put pollen, bee pollen in here, baking soda, very important, honey, a little bit of baking soda, and then a good spoon, a big spoon of pollen. Water. Pollen, water, honey, and lemon. 2 lemon, a lime. Today I put lime. They say it's good for you. Don't go one day without lime in water. Do not drink the lime juice straight. It's bad for your teeth. It's bad for your body. But you put it in a water or something like that, and 40 minutes, all you have is 40 minutes to drink it. It has a lot of great effect on your body. I don't know. I read these things. I listen to these things from books, and then I just do it. Anyhow, glycolysis. Yes, we will talk about that. Oxidation of pyruvate. Remember oxidation? We talked about this. When a molecule loses electron, sodium underwent oxidation. You will see that in glucose. The citric acid cycle, electron transport chain, I guess they're in the sequence of events that are energy yield, aerobic respiration, regulation of aerobic respiration, regulation of aerobic respiration. Very good. Oxidation without O2, you will see that. Alcohol is one of them. At home, when you make beer or you make wine, this is it. No oxygen is supposed to get into your apparatus. Anyhow, catabolism of proteins and fats, definitely very important. And evolution of metabolism, it's okay. At the beginning, everything was glycolysis, all of the cells, but the oxygen came later on for respiration. Anyhow. Respiration. Organisms can be classified on how they obtain energy, autotroph and heterotroph. You possibly know this from middle school, elementary school. Autotroph are organisms that they make their own. Auto, it means self, right? Auto, it means self, and troph, it means food, feed. So they feed themselves. Example, plants. You saw in the lab serratium. Okay, some of these spirogyra. Okay, these are some able to produce their own organic molecules through photosynthesis, which you did it in the lab. Heterotrophs, on the other hand, talking about us. We cannot make our own food. No way. We need to eat it from outside, from plants. Okay, live on organic compounds produced by other organisms. All organisms use cellular respiration to extract energy from organic molecules, even plants, even bacteria. They use cellular respiration, one form or another. You're going to learn two or three of them. I used to talk about the other one, but anyhow, two of them at least. We'll see what comes up. Two of them at least. The one that needs oxygen, the one that do not need oxygen. Even bacteria do that. Bacteria do cellular respiration. Viruses don't do that. We don't consider viruses as living organisms. They're not living organisms. They do not do respiration. Any kind of respiration we know of. None, zero, zilch. Viruses, bacteria do. Okay, cellular respiration is a series of reactions: oxidized, lose of electron, and reduced, gain of electron. We talked about all of this. Chlorine. I'm saying it in simple forms that you understand it now, and then things are going to become a little bit not complicated. It's very simple, straightforward, but the molecules are going to become a little bit bigger. Sodium and chloride, they're just two elements. They exchange electrons. So chlorine is a reduce, gain electrons. It's being reduced. But anyhow, dehydrogenation, it means lost electrons are accompanied by protons. Yes, a hydrogen atom is lost one electron and one proton. A hydrogen atom loses, you will see it when hydrogen atom loses its electron, then it would have, because hydrogen has only one electron, right? When it loses its electron, then it becomes just a proton, right? You remember that from the beginning of semester. So redox, we talked about this again. Let's talk about it. During redox reaction, electrons carry energy from one molecule to another molecule. In here, we have an intermediate molecule to accept the electrons. That intermediate molecule is NAD. What does NAD stands for? Nicotinamide adenosine dinucleotide. I hope the picture comes up. I hope so. So when NAD accepts an electron, it becomes NADH. Okay, so there is a molecule here breaks. Molecule here, glucose, I should say it because it's coming up soon. Breaks. When it breaks, it gives off electron. Oxidizes. Breaks. Remember, anytime you guys forget the term oxidize, think of a piece of a metal that is out in the rain, and out there it will rust. It is breaking. I hope you've seen some rust at something. It's breaking. Okay? So glucose molecule get oxidized and break. When it's breaking, it gives off its electrons to whom? Right there, NAD. Okay? So nicotinamide, I'm going to say NAD from now on. Nicotinamide, adenosine dinucleotide, the picture might come up and not. They give you the structure of it. An electron carrier, good definition. Electron carrier for enzymatic cofactors. You'll see. So NAD accepts 2 electrons and 1 proton to become NADH from breaking down of glucose molecule. Reaction is reversible, yes, but different part of the cell. Where this is happening when NAD becomes NADH is in this part of the cell, you will see. And when NADH becomes NAD, it's in this part of the cell. You will see. It is reversible. Yeah, but different enzymes. The judge and the priest. 2 different people. Different enzymes are involved. Okay, what is he talking about here now? Oxidation when the molecules go this way, reduction when the molecules go this way. When you have a high energy molecule like glucose molecule goes sit here, and then it converts NAD to NADH. Okay, enzymes that use NAD as a cofactor for oxidation reactions binds to NAD and the substrate. In oxidation reduction reaction, electrons and a proton are transferred NAD to NADH for a second proton is donated to solution as well. I'm not too crazy about all of what is going on in here. Yeah, NAD, the main thing is this one. So the NADH can become an NAD as well. NADH diffuses away and can be donate electrons. Yeah, this is important, but I'm not too crazy about it. Energy harvest and redox reaction. In overall cellular energy harvest, dozens of redox reactions takes place. You will see that. Do not worry about the number dozens. He's just saying a lot of chemical reactions occur. You will see that. Number of electrons acceptors, including NAD, yes. In the end, high energy electrons form initial chemical bonds and have lost much of their energy, transfers to a final electron acceptors. You will see all of this. You will see. I hope with diagrams. Electron acceptors. So aerobic respiration, it means oxygen is involved. AER in your root words means air. Right here, oxygen involved in this reaction. And you will see it. Okay. Anaerobic, AN in front of a word means what? Without. Air, without air. No oxygen involved. The final acceptance is the inorganic molecule, which you see it, not O2. Fermentation, yes, final electron acceptor is an organic molecule, which you will see that fermentation, you saw it with yeast. We didn't look at yeast, but making bread, if you're making bread at home with yeast, this is the fermentation is okay. You're making beer at home. You're making wine at home. All of that is fermentation. Okay, so this is the overall reaction for cellular respiration. I would like you to know this. C6, a glucose molecule has how many carbon? 6. How many hydrogen? 12. How many oxygen? 6. Plus oxygen, it gives you carbon dioxide. That's what you're exhaling. Water molecule and energy. Okay, so the free energy, the delta G, remember that? The delta G you get is minus, it's, of course, is an exergonic reaction. Remember that? Cellular respiration is exergonic reaction. They're releasing energy. The releasing energy of glucose molecule is minus, of course, now you know why, 686 kilocalorie per mole. So free energy can be even higher than this in a cell. So this large amount of energy must be released in small steps rather than all of the sudden. It is released step by step. If it is released all of the sudden, the cell burns. So that's why the cell has to go through steps to release that energy. Okay. So electron from food, it goes step by step by step by step by step. And of course, at the end, with the help of oxygen and the hydrogen, it becomes water. Electron carriers, many types of carriers used, soluble, membrane-bound, move within the membrane. All carriers can be reversibly oxidized and reduced. Some carry just electrons, some electrons, and protons. NAD requires 2 electrons and a proton to become NADH. You will see that. I will talk about, this is not that significant to me, okay? So, but when I show you the diagram, hopefully things will make a little bit more sense. All of these will make sense. Right now, you know, okay, great. They did show you the nicotinamide molecule. This is the nicotinamide, NAD, just the NAD itself. Look, right here is a sugar molecule. Here is adenine. Remember that? And here is the couple of phosphate. And here is another sugar molecule. And here is the nicotine. So, and this is NAD. Very good. They're showing you NAD. Look at the hydrogen here. Wait. This one. Sorry about that. Look at this one right here. One hydrogen. All right. Now, when he accepts one more hydrogen, becomes double right here. And then this bond becomes single. Remember, this is carbon, and here is carbon, and carbon must have 4 covalent bonds. 1, 2, 3, 4. This one, 1, 2, 3, 4. In both cases, carbon has 4 covalent bonds. This is the NADH right here. This is NAD. Where that H came from? Glucose molecule in my lemon, in the lemon that I drank. Okay, I hope I'm making some sense. So they did show you the diagram. So ATP, cells uses ATP to drive endergonic reaction. Yes, and then you will see that in photosynthesis as well. Delta G free energy of hydrolyzing terminal phosphate is minus 7.3 kilocalorie. And then 2 for ATP. Two mechanisms of synthesis, substrate level phosphorylation. I think we talked about it here. When you have a molecule and you add this is phosphate. You add phosphate to it, it's called phosphorylation. Remove the phosphate, it's called dephosphorylation. We talked about it with GTP, the G proteins, and tyrosine kinase. Adding phosphate is phosphorylation. Removing phosphate is dephosphorylation. So transfer phosphates groups directly to ADP during glycolysis. You will see that he's putting a lot of words. I wish he would come up with the diagram so I can talk about it a little bit more, but that's okay. So oxidative phosphorylation, ATP synthase. The enzyme that makes ATP is called ATP synthase. You remember at the end of the word ACE, it means enzyme. So use energy from a proton gradient. You will see that in mitochondria. I guess you have to put all of these write-ups at the end when the pictures come up. Things will, I hope. If it doesn't, I hope you don't ask me the day of the exam. Just like this morning. I'm not saying it's good that you asked me this morning questions before the exam. I loved it. I enjoyed it. Number one, because I see you're in the class, you're involved, but it would be nice to ask them a little bit before the exam. So for the exam, you're ready to go. Ask me anytime, even before the exam. But anyhow, so you will see that. Okay, substrate level phosphorylation. We talked about this, you guys. Oh, he's talking about PEP, phosphate, adenosine, ADP. So pyruvate. He hasn't talked about glucose molecule breaks down to pyruvate yet. Come on. The complete oxidation of glucose molecule proceeds in stages. Very good. The first step of cellular respiration is glycolysis. Omar asked that. In that order. You should know it in that order. You do not have quiz next week, but in 2 weeks from today, you have a quiz over these chapters. The complete oxidation of glucose proceeds in stages: glycolysis, pyruvate oxidation, citric acid cycle, and electron transport chain. You will see them step by step with pictures. If it doesn't have pictures, then I'll go get the pictures. Here we go. Oh, okay. This is aerobic respiration. It means it needs oxygen. Here's a glucose molecule. It breaks down to pyruvate. Pyruvate goes inside of the mitochondria. But this glycolysis, the definition of glycolysis is breakdown of one glucose molecule to 2 molecules of pyruvate. That's glycolysis. When one glucose molecule breaks down to 2 molecules of pyruvate, it is glycolysis. Okay? And then pyruvate goes to mitochondria and breaks down even more. And that is when you really get energy. You remember last exam today you took? Mitochondria is the power plant of the cell. It gives you a lot of ATP. How, Amir? Right now, right here. Okay? And all of these steps, all of these 4, sorry about that, 4. Right here is glycolysis, pyruvate oxidation becomes acetyl-CoA, citric acid cycle or Krebs cycle. The first scientist who won the Nobel Prize for discovering this was Heinz Krebs. They call him citric acid cycle in this diagram, a Krebs, K-R-E-B-S, Krebs cycle. And after that is the electron transport chain right here. The 4 stages are right here. These 4 stages, they showed it to you in a diagram. Glycolysis. Oh, come on. Pyruvate oxidation. Citric acid cycle. And of course, the last one is, oh, come on, electron transport chain. Okay. So glycolysis is conversion of one glucose molecule six carbons to 2 molecules of pyruvate, 3 carbons. So 2 times 3, last time I checked my calculator, was 6. 10 steps. Breakdown of 1 glucose to 2 pyruvate has 10 steps. Occurs where? In cytoplasm. Remember I said this happens this side of the cell, this happens this side of the cell. You have to know these things. Where does it happen, glycolysis? In the cytoplasm of the cell. If you study these PowerPoints, you listen to these videos, you're coming to the quiz in 2 weeks prepared. You don't, you're coming there hoping to know the answer. Hoping. Net production of 2 ATP molecules by substrate level phosphorylation. So you will see there are 4 ATP generated. That's a gross, G-R-O-S-S. But 2 of them were invested. So at the end, you get 4. So that's what he's saying. When you get your paycheck, you remember that, you guys? When you get your paycheck, there is a gross and there is a net. Uncle Sam, Social Security, Medicare, whatever. The state of California, they take their share. And then you get a net pay. So there is a gross pay, there is a net pay on your paycheck. And 2-NADH also produced. 2-NADH, gross 4 ATP, net 2 ATP from breakdown of 1 glucose molecule to 2 molecules of pyruvate. Let's go. So here's a glucose molecule with 6 carbon. It breaks down to 6 carbon sugar diphosphate. He didn't mention every steps. It's okay. You don't need to know them. And then it breaks down to 2 pyruvate molecule at the end. What happens? You had to invest 2 ATP to break down this 6 carbon together. When you invest 2 ATP at the end, when pyruvates are formed, you made 4 ATP. I talked about that. And of course, you made 2 NADH as well. Everything in there you should know. So it's okay. He's mentioning where the ATPs are coming up step by step, the 10 steps. You remember I said from breakdown of glucose molecule, come on, go up, please. From breakdown of glucose molecule to 2 molecules of pyruvate, there are 10 steps, and he numbered them for you. Step number 1, glucose becomes glucose 6-phosphate. ATP becomes ADP. So the name of the molecule is fructose 6-phosphate now. There is an enzyme involved here, hexokinase. There is an enzyme involved here to break down glucose 6-phosphate to fructose 6-phosphate. It's called phosphoglucose isomerase. There's an enzyme involved to make fructose 6-phosphate to fructose 1,6-phosphate. The second ATP comes into picture. Remember, you have to invest, you have to give these 10 steps to ATP. Anytime in biology, I said it long time ago. Even before you start taking this class, I said it. Anytime you want to break a bond, a chemical bond in biology, you need energy. You're breaking the bond between the covalent bond between the carbons of glucose molecule. 2 ATP needed. And then, of course, the rest, a few chemical reactions occur. Name of the enzymes are mentioned here, everything mentioned here. And then at certain steps, you make ATP, ADP is converted to ATP, ADP is converted to ATP. And then, of course, water is released, etc., etc., etc. All of the details you don't have to worry about. It's just showing you a lot of details, which is okay, which is good. But I want you to know, first of all, the name of the process. I already talked about all of these, but let's recap. I think this is the last slide. Yeah, this is the last slide. This process happens in cytoplasm. Glycolysis. Glycolysis is breakdown of 1 glucose molecules to 2 molecules of pyruvate. There are net 2 ATP formed, gross 4 ATP formed. And for NADH, we don't have gross and net anymore. Just 2 NADH is formed. 2 pyruvate is formed. 10 steps. That's it. You have any question, please ask. Okay, NAD must be regenerated. For glycolysis to continue, NADH must be recycled to NAD by either aerobic respiration or fermentation. Oxygen is available, and the first electron acceptors produce significant amount of ATP. You will see that. All of this will come into picture. You will understand all of this at the end. But he's giving you good information. You have to go through all of these. You have to listen to all of these lecture points, and you will come and study each one of these. Will make sense. Fate of pyruvate depends on oxygen availability. When oxygen is present, pyruvate is oxidized, yes, to acetyl-CoA, which enters a citric acid cycle, aerobic respiration. Without oxygen, pyruvate is reduced to, in order to oxidize NAD back to NAD fermentation. Yeah, you will see that. Okay, so far, glucose broke down to 2 pyruvates. Now what happens? Pyruvate moves and goes into mitochondria. Okay, pyruvate without oxygen. Oh, he's talking about, okay, there is no oxygen in some bacteria when you're making wine. Okay, so it goes this route. It goes to acetaldehyde, and then from acetaldehyde, it goes to ethanol. When you are making bread, this carbon dioxide make your dough to rise. And you're using yeast. Yeast is doing the fermentation for you. Okay? Then when there is oxygen involved right here, pyruvate is turned into acetyl-CoA. And of course, when pyruvate is turned into acetyl-CoA, you need NADH. NAD becomes NADH. Another NADH is made for you. And then the acetyl-CoA goes to the citric acid cycle. this is confusing. He shouldn't done this, your textbook. From conversion of pyruvate to acetyl-CoA, you really do not need oxygen. Even the citric acid cycle, this one does not need oxygen. It's just the electron transport chain down here. It needs oxygen. He's saying, well, this route, you do need oxygen. Yeah, but later on the bottom right here. Oh, another one, when you exercise lactate, he's jumbling up all of these together. When you go exercise and you have pain and ache, you have not been running, so your body forms lactic acid around your muscles, so you have aches and pains. And that is that, the lactate. It happens in your muscle cells. No acetaldehyde is formed. Okay, he's jumbling all. Pyruvate oxidation, here we go, occurs in mitochondria of eukaryote, multi-enzyme complex called pyruvate dehydrogenase catalase enzyme. Yeah, this is good. I don't expect you to know this, but remember I said vitamins? This enzyme is a huge enzyme. It's a huge, they call it complex. It needs vitamins. Your B vitamins are needed here. Some of them, I can't remember. I took these courses a long time ago. So your vitamin Bs are needed there. And occurs in plasma membrane of prokaryotic cells. Products of pyruvate oxidation from each 3 carbon pyruvate molecules, 1 carbon dioxide decarboxylation by pyruvate dehydrogenase. Yes, he's giving you the name of the enzymes. I don't expect you to know the name of the enzymes. But I would like you to know you lose 1 carbon dioxide when pyruvate becomes acetyl-CoA. Also, you make 1 NADH right here. 1 acetyl-CoA, which has 2 carbons from pyruvate attached to coenzyme A. But that's what they call it, acetyl-CoA, because it's a coenzyme, another vitamin involved here. Coenzyme A. Acetyl-CoA proceeds to citric acid cycle if the cell needs ATP. The cell does not need ATP, this becomes fat. We will see. If you do not study for your biology class, this acetyl-CoA turns into fat. You study for your biology class, this becomes energy. So you can think, you can study. Let's go. Oxidation of pyruvate. So pyruvate oxidation, it does not need oxygen, but it turns and it becomes acetyl-CoA. And of course, it releases a carbon dioxide because pyruvate has 3 carbon. Acetyl-CoA has 2 carbon plus a huge molecule here called coenzyme attaches to these 2 carbon. I hope they have a picture of the CoA. Here we go. Yeah, they might have a, I used to have a picture. I will bring it up from another class. But anyhow, you see the S is sulfur attaches to the CoA. The S of the CoA attaches to the 2 carbons of, now this molecule is called acetyl-CoA. And of course, this is pyruvate. It loses this CO2. Okay. Let's stop here. And so next time I will talk about the citric acid cycle, and that would be the end of this. I haven't put these lectures in YouTube videos. I will. This is chapter 7, chapter 6, and this will be on the chapter 7. Okay. So today's video, I talked about a little bit about chapter 6 as well. But Monday video, I put it on, oh, come on, stop sharing. Okay, the Monday video, I put it on chapter 6, and today's I put it on chapter 7. Right.